Afternoon class. Today we're going to continue Unit 7. This is Chapter 24, Part B, Nutrition, Metabolism, and Energy Balance. 24.6, Protein Metabolism. So we've looked at carbohydrate metabolism, we've looked at lipid metabolism. So what is the fate of proteins? So proteins deteriorate, so they need to be continually broken down and replaced. So proteins, imagine the ones that are embedded in the cell, the plasma membrane, enzymes and in the lysosomes, for example, or enzymes all over inside the cell or at specific sites on the membranes. So there's lots of proteins, and they break down, and so they have to be replaced. The amino acids are replaced into new proteins or different compounds, and proteins are not really stored in the body. Unfortunately, we know that carbs are stored glucose is stored in the form of glycogen, and lipids are stored in adipose tissue, but proteins are not. So when dietary proteins are in excess, amino acids are oxidized for energy, or they're converted to fat for storage. The goal is to produce molecules that can be used for energy in the citric acid cycle or converted to glucose. So just like lipids, we don't have a separate pathway for proteins, for the breakdown of proteins. So the first step in amino acid breakdown is deamination, whereby the NH2, the amine group, is removed, and then it's converted to pyruvic acid, or one of the keto acid intermediates of the citric acid cycle. So there are three events of the amino acid degradation. First one is transamination, second one is oxidative deamination, and the third one is keto acid modification. So let's look at transamination. So many amino acids can transfer their amine group to alpha ketogluteric acid, transforming it into glutamic acid. So as a result, the original amino acid becomes a keto acid, and glutamic acid is an intermediate keto acid of the citric acid cycle, and this reaction is reversible. The second is oxidative deamination. In the liver, the amine group of glutamic acid is removed as ammonia, and then alpha ketogluteric acid is regenerated. The ammonia then combines with CO2 to form urea and water, which is a less toxic form of a nitrogenous waste. Urea is released to the blood, and then it's excreted in urine, and this urea cycle helps rid the body of toxic ammonia. The third step is the keto acid modification. So keto acids are formed from transamination. They're altered to produce metabolites that can enter the citric acid cycle. So major metabolites produced are pyruvic acid, acetyl-CoA, alpha ketogluteric acid, and oxaloacetic acid. If you remember, these are all part of they all enter the citric acid cycle. You know how pyruvic acid gets converted to acetyl-CoA combines with oxaloacetic acid, and then alpha ketogluteric acid is one of the intermediates of the citric acid cycle. Glycolysis reactions are reversible, so pyruvic acid metabolites formed can be reconverted to glucose, and this contributes to gluconeogenesis, which we've talked about. In other words, production of glucose from non-carbohydrate sources. So this figure, 2417, summarizes what we've seen so far. Okay, so we have here in the liver the amino acid plus a keto acid, like alpha ketogluteric acid. We have transamination, where an amine group is switched from an amino acid to a keto acid. And then we have, in the second step here, oxidative deamination. The amine group of glutamic acid is removed as ammonia and combined with CO2 to form urea. Urea goes into the blood, and then the kidneys will filter the blood, and will filter the urea, which is excreted in urine. The keto acid, if we go back here, so keto acid modification, the keto acids formed during transamination are altered so they can easily enter the citric acid cycle. Quick summary of amino acid catabolism is seen in figure 24.18. So transamination is it's transferring an amine group from an amino acid to alpha ketoglutarate acid, generating glutamic acid. And oxidative deamination is the removal of the amine group in the form of ammonia from the glutamic acid and regenerating alpha ketoglutarate acid. Amino acids are most important anabolic nutrients. They form all proteins as well as the bulk of the functional molecules. Protein synthesis that occurs on the ribosomes is hormonally controlled. For example, growth hormone, thyroid hormone, and sex hormones, such as estrogen, estradiol, progesterone, and testosterone. Protein synthesis requires the complete set of amino acids, and we already talked about essential amino acids that must be taken in from our food. During our lifetime, depending on the body size, we can synthesize about 500 to 1,000 pounds of protein. Okay, so that concludes that aspect of metabolism. Now we're going to talk about metabolic states of the body. So this is in section 24.7. Catabolic-Anabolic steady state. So there's a dynamic state in which organic molecules, except for DNA, that are continuously broken down and rebuilt. So body uses these nutrient pools, which are stores of amino acids, carbohydrates, and fats. And the pools are interconvertible because pathways are linked by common intermediates, right? We said there's that common pathway there, the glycolysis Krebs cycle, that links all of these molecules. The amount and direction of conversion are directed by the liver, adipose tissue, and skeletal muscle. Let's start looking at the amino acid pool. So this is the body's total supply of free amino acids. So we lose proteins in urine, our hair, and skin cells, and they're typically replaced by our diet. And the pool is the source for resynthesizing body proteins, forming amino acid derivatives, and gluconeogenesis. Regarding carbohydrate and fat pools, so carbohydrates and fats are easily interconverted through key intermediates. There are two major differences between carbohydrate fat pools and amino acid pool. Fats and carbohydrates are oxidized directly to produce energy. Amino acids must first be converted to a citric acid cycle, keto acid, and any excess carbohydrate and fat can be stored as such. Amino acids are not stored as proteins. Figure 24.19 shows the interconversion of carbohydrates, fats, and proteins. Okay, so let's start with the middle. Okay, let's start with carbohydrates. Okay, so glycogen is the storage form of glucose. So glycogen can be converted to glucose, and glucose can be converted to glycogen. And glucose can, when phosphorylated or primed, it becomes glucose 6-phosphate. It's converted to glyceraldehyde 3-phosphate, which becomes pyruvic acid. And this, of course, is reversible. And then once we have pyruvic acid, it gets converted to acetyl-CoA, and this can enter the citric acid cycle. So we know this, hopefully. Okay. With proteins, the proteins get broken down into amino acids. and amino acids are used to make proteins that are needed by the body. But remember, amino acids are converted to keto acids, which enter glycolysis by being converted into the intermediates of glycolysis. But also the amino acids are deaminated and become NH3 or ammonia, which gets converted into urea that is excreted in urine. But those keto acids, they're also, they can become intermediates in the citric acid cycle, and if needed, they can be converted back into keto acids. For fats, so triglycerides are broken down into glycerol and the fatty acids, and these can be diverted into intermediates of glycolysis to become pyruvic acid, and also intermediates to become acetyl-CoA that enters the citric acid cycle. So any excess is that the body would use would become ketone bodies, and pyruvic acid, if you look here, can be converted to lactic acid in, we said, anaerobic conditions when there's no oxygen. So let's look at absorptive state. This is also called fed state, and which lasts for approximately 4 hours after eating, when the absorption of nutrients is occurring. And at this point, anabolism exceeds catabolism. So excess nutrients are stored as fats if not used. So for carbohydrates, glucose is the major cellular energy fuel, and it can be converted in the liver to glycogen or fat. The glycogen remains in the liver, but fat joins protein to form very low density lipoproteins, VLDLs, that are released into the blood for storage by adipose tissue. Blood-borne glucose enters the cells, and any excess is stored as glycogen in muscles or fat in adipose cells. So what about triglycerides? Chylomicrons from lymph are hydrolyzed to fatty acids and glycerol before passing through the capillary wall. Lipoprotein lipase is the enzyme that catalyzes the hydrolysis of lipids, and this enzyme is abundant in muscle and fat tissues. Triglycerides are used by adipose tissue, the liver, and skeletal and cardiac muscle as the primary energy source. Most glycerol and fatty acids are converted back to triglycerides for storage. For amino acids, so some absorbed amino acids are deaminated in the liver to become keto acids that can be used in the citric acid cycle. Any excess amino acids can be stored as fat in the liver. Most amino acids are taken up by the cells and used for protein synthesis. Figure 24.20 shows the major events and principal metabolic pathways of the absorptive state. Okay, so let's look at the, so on the left, major metabolic thrust, anabolism and energy storage. So amino acids are used to make proteins. Glucose is stored as glycogen, and glycerol and fatty acids are converted to triglycerides. Major energy fuel, glucose from the diet, gets converted to, broken down to CO2, water, and ATP, as we know. And at the level of the liver, amino acids are deaminated and used for energy or stored as fat. So the amino acids are converted to keto acids, and then any excess stored as triglycerides, or if needed, used up for energy to generate CO2, water, and lots of ATP. In figure 24.20, part B, major events. So this here, principal pathways of the absorptive state. Okay, so from the gastrointestinal tract, glucose is absorbed, gets converted to glycogen in muscle, and then in muscle, yes. Okay, so again, if we go back to the gastrointestinal tract, it absorbs the amino acids that get converted to protein, like muscle protein, or they can be used in the liver to make proteins, converted to keto acids for energy, and then the keto acids are converted to fatty acids to make triglycerides. The glucose, again, that is absorbed, it doesn't only get converted to glycogen, but it also gets converted to glyceraldehyde 3-phosphate, and also which is can be converted to glycerol, which combines with the fatty acids to make triglycerides. The glucose also in adipose tissue and fat tissue gets converted to fatty acids and glycerol to make triglycerides that are stored in adipose. From the gastrointestinal tract, we have triglycerides, which get stored in adipose tissue. So they're broken down into the glycerol that combines with the fatty acids to make triglycerides, as seen here. And if we go back to the gastrointestinal tract on the right, the absorption of glucose also is used up in all tissues to make energy, to make ATP. So glucose is very versatile because you can use it for energy, you can use it for fat storage, and you can use it to be stored as glycogen. Hormonal control of the absorptive state. So the absorptive state is controlled primarily by the hormone insulin, which is produced by the beta cells of the pancreas and is stimulated by elevated blood glucose levels of glucose and amino acids. Intestinal GIP, or glucose-dependent insulinotropic peptide, and parasympathetic stimulation as well. When insulin binds to membrane receptors, it facilitates the diffusion of glucose into the muscle and adipose cells. And the brain and liver take up glucose without the help of insulin. Insulin is a hypoglycemic hormone that enhances glucose oxidation for energy. And glycogen and triglyceride formation, and the active transport of amino acids into the tissue cells, as well as protein synthesis. So anything that lowers blood glucose levels, basically, that's the role of insulin. It also inhibits glucose release from the liver and gluconeogenesis. In other words, you don't want more glucose to be released into the blood when already its levels are elevated. Figure 24.21, insulin directs nearly all events of the absorptive state. Okay, so the stimulus is high blood glucose levels, and this will stimulate beta cells of the pancreatic islets to increase blood insulin. So it targets tissue cells and will increase the active transport of amino acids into tissue cells, which through cellular respiration be broken down for energy. And it also will increase the facilitated diffusion of glucose into the tissue cells for protein synthesis. So this active transport of amino acids into tissue cells can also enhance glucose conversion to fatty acids and glycerol, and as storage into glycogen. So if we look back at the increased active transport of amino acids into the tissue cells, this acts as a negative feedback loop where falling glucose levels will shut off the initial stimulus, and then no more insulin is going to be produced. Homeostatic imbalance 24.3. So you've probably all heard of diabetes mellitus. There's usually at least one person in the family where there's an inadequate insulin production or abnormal insulin receptors, which results in unavailability of glucose to most body cells. So basically, you have all this glucose, these high elevated levels of glucose in the blood, and the glucose can't enter the body cells, can't be available to the body cells. So it's excessively high blood glucose levels as a result, because the glucose doesn't get used up for energy or for other storage, etc. And there's glucose that is lost in the urine. So because the body needs energy, it will use fats and proteins for energy. And this can lead to metabolic acidosis, protein wasting, and weight loss if not treated. Okay, so now let's look at post-absorptive state. So post-absorptive state is also called fasting state. This is when the GI tract is empty and energy sources are supplied by breakdown of body's reserves. So such as the catabolism of fat, glycogen, and proteins would exceed anabolism. The goal is to maintain blood glucose between meals by making glucose available to the blood and promote the use of fats for energy. So glucose sparing saves glucose for organs that need it the most, such as the brain. So what are some of the sources of blood glucose? So first, glycogenolysis in the liver is the first reserve that is used. Glycogenolysis in skeletal muscle. Before glucose from the liver is exhausted, glycogen stores in the skeletal muscles start to break down to release glucose. Lipolysis in adipose tissues and liver. So the glycerol is used for gluconeogenesis in the liver. And catabolism of cellular protein. This is the last resort. So it's a major source during prolonged fasting, where there's a limited amount of protein that can be broken down before damage. So the amount of fat in the body determines how long a person can survive without food. Glucose sparing. So during prolonged periods of fasting, the body uses more non-carbohydrate sources to conserve glucose. So more fat and protein are converted to intermediates that can enter the citric acid cycle. The brain uses the bulk of the glucose, while other body cells will switch to fatty acids as a fuel source. And this results in the production of ketone bodies. After 4 to 5 days without food, the brain will start to also use ketone bodies as the energy source. Figure 24.22, major events and principal metabolic pathways of the post-absorptive state. Okay, so the major metabolic thrusts are catabolism and replacement of fuels in the blood. So proteins get converted to amino acids, glycogen gets broken down to glucose, triglycerides will be converted to glycerol and fatty acids. So the major energy fuel, so glucose provided by glycogenolysis and gluconeogenesis, fatty acids and ketones. Okay, so glucose is broken down into energy as well as the fatty acids and ketones. At the level of the liver, the amino acids are converted to glucose through gluconeogenesis. Remember what that means? It means making glucose from non-carbohydrate sources. And the amino acids are converted to keto acids, which then are converted to glucose. Figure 24.22b, principal pathways of the post-absorptive state. Okay, so where do we start here? So glucose that is stored in the form of glycogen is released into the blood and is used up principally by the nervous system, the brain, for fuel. Then glucose is it's converted from glycogen into pyruvic acid and lactic acids, and these make glucose. And then the triglycerides in adipose tissue get converted into glycerol and fatty acids. Fatty acids break down into ketone bodies, which are used up as fuel, and also the glycerol is converted to glucose. The fatty acids and glycerol from the triglycerides and the ketone bodies would be used by most tissues for fuel to make ATP. In muscle, the proteins are broken down into amino acids. Amino acids are converted in the liver into the keto acids, which get converted to glucose. Hormonal and neural controls of the postabsorptive state. So the sympathetic nervous system interacts with several hormones to control the events of the postabsorptive state. So it's more complex than the absorptive state, which utilizes insulin as the one hormone. The post-absorptive state is triggered by reduced insulin release as blood glucose levels drop. The main hormone in the post-absorptive state is glucagon, which has the opposite effect as insulin. It is hyperglycemic, and its release is stimulated by declining blood glucose levels, rising amino acid levels, and glucagon promotes glycogenolysis and gluconeogenesis in the liver and lipolysis in adipose tissue, causing the fatty acids and glycerol to be released. What happens after a high protein, low carbohydrate meal? So insulin responds to high amino acid blood levels. If high protein meal is consumed, hypoglycemic conditions may result, which is not good for the brain, because we said the brain always needs this continuous supply of glucose. So glucagon modulates the effect, ensuring there's always an adequate blood glucose supply. So figure 24.23 shows the effect of glucose as a hyperglycemic hormone that stimulates a rise in blood glucose levels. So falling plasma glucose and rising amino acid levels stimulate the alpha cells of the pancreatic islets to increase plasma glucagon, which stimulates glycogenolysis and gluconeogenesis in the liver, which will increase plasma glucose levels. Now, plasma increases in plasma glucagon levels will stimulate lipolysis in the adipose tissue, which is going to increase plasma fatty acids, and the fat is used by the tissue cells, and this is called glucose sparing. So this increase in plasma glucose is going to act as a negative feedback mechanism. So rising glucose levels will shut off the initial stimulus. So the sympathetic nervous system, also known as the fight or flight nervous system, so the adipose tissue is innervated by the sympathetic nervous system, and it can quickly supply glucose if blood levels are low. So low plasma glucose, fight or flight response, or exercise can trigger fat mobilization and glycogenolysis. And this is initiated by the sympathetic nervous system and epinephrine that is released from the adrenal medulla, which is part of the adrenal gland. It's the inner core of the adrenal gland. Other hormones, metabolism and nutrient flow are also influenced by hormones other than glucagon and insulin, such as growth hormones, thyroxine that is produced by the thyroid gland, sex hormones, and corticosteroids can have an effect on metabolism, and most of their effects are indirect. Table 24.4 summarizes all the hormones that influence metabolism. So if we look at insulin, it stimulates glucose uptake by cells, and thyroxine as well. Amino acid uptake by cells is stimulated by insulin as well as growth hormone. Glucose catabolism for energy is stimulated by insulin and thyroxine. Glycogenesis is stimulated by insulin, as well as lipogenesis and fat storage, and gluconeogenesis is inhibited. Insulin also stimulates protein synthesis, as well as growth hormone, thyroxine, and testosterone. The glycogenolysis is stimulated by glucagon, epinephrine. Lipolysis and fat mobilization is stimulated by glucagon, epinephrine, growth hormone, thyroxine, and cortisol. gluconeogenesis is stimulated by glucagon, epinephrine, growth hormone, and cortisol. And cortisol stimulates protein breakdown. For these, basically, you should know the effects of insulin and glucagon. Those are the I don't expect you to remember all the other ones. 24.8, metabolic role of the liver. So the hepatocytes, or liver cells, carry out over 500 metabolic functions. They process nearly every class of nutrient, and they play a major role in regulating plasma cholesterol levels. They also store vitamins and minerals. They metabolize alcohol, drugs, hormones, and bilirubin. Table 24.5, this is a summary of metabolic functions of the liver. So for carbohydrate metabolism, and it's basically, the main job is to maintain blood glucose homeostasis. So the liver is going to convert galactose and fructose to glucose, and it stores glucose in the form of glycogen when blood glucose levels are high, in response to hormonal controls. It performs glycogenolysis and releases glucose to the blood. Gluconeogenesis, it converts amino acids and glycerol to glucose when glycogen stores are exhausted and blood glucose levels are falling, and converts glucose to fats for storage. Then there's fat metabolism. You can read through it. It's a nice review. Protein metabolism as well. What's new is vitamin mineral storage. So the liver stores vitamin A for about 1 to 2 years supply. It stores sizable amounts of vitamins D and B12, about 1 to 4 months supply. And it stores iron. Other than iron bound to hemoglobin, most of body's supply is stored in the liver as ferritin. In terms of biotransformation functions, it metabolizes alcohol, drugs, and other toxins by inactivating them for excretion by the kidneys, and performs reactions that may result in products which are more active, changed in activity, or less active. It also processes bilirubin resulting from the breakdown of red blood cells and excretes bile pigments in bile. And it metabolizes blood-borne hormones to forms that can be excreted in urine. So cholesterol metabolism and regulation of blood cholesterol levels. Cholesterol is a steroid, if you remember, is not used as an energy source. It is a structural basis of bile salts, steroid hormones, and vitamin D. And it's also one of an important component of plasma membranes. 15% of blood cholesterol is ingested, with the rest which is made in the body primarily by the liver. And it's lost from the body when catabolized or secreted in bile salts that are lost in feces. Cholesterol is a steroid, so it's not water soluble. So how is it transported? You have lipoproteins that transport water insoluble cholesterol and triglycerides through the blood. They regulate lipid entry and exit at target cells, and all contain triglycerides, phospholipids, cholesterol, and protein. The higher the percentage of lipids, the lower the density. There are types of transport lipoproteins, HDLs and LDLs. HDLs are high-density lipoproteins. These have the highest protein content. So the LDLs are also, they're a low-density lipoprotein, have the highest cholesterol content. The VLDLs means very low-density lipoproteins. The contents are more than half triglycerides with low density of proteins. And chylomicrons, also known as ULDLs, or ultra-low density lipoproteins, these will transport dietary lipids from the intestines to other parts of the body. They have the lowest density and consist almost entirely of triglycerides, up to 92% in fact. So again, these transport lipoproteins, it's because of the watery medium of plasma. So that's the way the lipids need to be transported. They need to be transported within these lipoproteins. Okay, so figure 24.24, composition and function of lipoproteins. Okay, so let's start with chylomicrons. These carry absorbed lipids from the intestine to the liver. So you see they consist mostly of triglycerides. Then the very low density lipoproteins. These are mostly triglycerides. And then low density lipoproteins. These don't have a lot of triglycerides, but they carry a lot of cholesterol. And the high density lipoproteins, these are mostly proteins. That's why they're called high density lipoproteins. And they're really small molecules. So if you look at one of them, what does it was? There's proteins, there's the phospholipid bilayer, cholesterol embedded in the membrane, but there could be cholesterol that's bound to the fatty acid within the lipoprotein, and there's triglycerides inside. So VLDLs transport triglycerides from the liver to peripheral tissues, mostly mostly to adipose tissue. LDLs transport cholesterol to peripheral tissues for membrane storage or hormone synthesis. And HDLs, these transport any excess cholesterol from the peripheral tissues to the liver to be broken down and secreted into bile, and also provide cholesterol to steroid-producing organs. So you've probably heard of HDL and LDL cholesterol. So what are the recommended total levels of these lipoproteins? So the maximum total cholesterol should be 200 milligrams per deciliter or less. Levels that are greater than 200 milligrams per deciliter are linked to atherosclerosis or hardening of the blood vessels. More important to measure form in which cholesterol is transported in the blood. So for example, HDL is thought to protect against heart disease because it takes cholesterol out of blood. Greater than 60 is good. Less than 40 milligrams per deciliter is not good. For LDL cholesterol, this will deposit cholesterol in the blood vessels. 100 or less is good, but anything above 130 is not good. Factors regulating blood cholesterol levels. So the liver produces cholesterol at a basal level regardless of dietary cholesterol intake. So restricting dietary cholesterol does not markedly reduce blood cholesterol levels, interestingly. More important effect is the relative amounts of saturated and unsaturated fatty acids. So saturated fatty acids, these stimulate the liver synthesis of cholesterol and inhibit cholesterol excretion from the body, whereas unsaturated fatty acids will enhance excretion of cholesterol into bile salts. So it's very important to eat foods that are that contain more unsaturated fatty acids. There's a couple of things that are really important here. One, cholesterol. Restricting cholesterol, is not the way to go. And the second thing is, and the other thing is, you've heard of, low-fat diets. I know it was a big thing in the 80s, but that was actually detrimental because if you eat very little fat, then, your body does not get rid of the cholesterol. So you need to just, it's very important to eat the right type of fat. Factors regulating blood cholesterol levels, continued. So trans fats, these can occur when healthy oils are chemically transformed into solids. An example of that is margarine. These will have a worse effect on cholesterol levels than the saturated fats. In fact, they increase the LDL and reduce the HDL. And Canada has, in the last 10 years, limited so it has asked the food industry to limit the amounts of trans fats in foods. Unsaturated omega-3 fatty acids found in cold water fish have lower proportions of saturated fats and cholesterol. These make platelets less sticky and help prevent spontaneous clotting. They also have anti-arrhythmic effects on the heart and can lower blood pressure. Stress and cigarette smoking have been shown to lower HDL levels. Aerobic exercise and estrogen increase HDL levels and decrease LDL levels. Also, body shape plays a role. You've heard of apple shape versus pear. So apple is when the fat is carried on the upper body, and this is correlated with higher cholesterol and LDL levels, whereas in a pear shape where you have the fat distribution mostly on the hips and thighs, and this is correlated with lower cholesterol and LDL levels. Clinical Homeostatic Imbalance 24.4. Risk factors for assessment of cardiovascular disease. So previously, high cholesterol and LDL-HDL ratios were used as predictors of potential disease, and these are not found to be as reliable. Now, LDL levels and other risk factors are believed to be more accurate. So treatment for high LDL levels are statins. These are cholesterol-lowering drugs, and there's about an estimated 2.5 million Canadians that take statins.